We are recording this. Just be with us. No lewd comments or anything like that. Hazel, don't say anything bad about anybody today. It's being recorded.
What?
So. All right, so here we go. The objective, we're just going to look at the structure of the cytoskeleton and the ECM, the extracellular matrix, and kind of relate their structure to their function a little bit. It's a little strange, but within the cytoskeleton, we also talk about cilia, flagella, vesicle movement, and cell membrane control. So can anybody tell me why we group cilia, flagella with the cytoskeleton? Cilia, flagella are outside. Why do we relate the two? Yes?
They both made of the same microfibers.
Very good. They're made of microtubules. They're made of similar materials, so they all get kind of grouped together. Did I walk through the walking proteins with you guys before?
Yeah.
Okay, good. So you have a little bit of contact. You're a little bit ahead of the other class. So just to kind of recap, peroxisomes, right, they help to detoxify the cell. We're good, just to make sure we're doing peroxisomes to get it out of the way before we move forward. Peroxisomes, they work in conjunction with mitochondria and chloroplasts. Everyone okay with peroxisomes? We should be good with our organelles. So cytoskeleton, central cilia, and flagella. Cytoskeletons are microtubules, microfilaments. So I've already gone through this with you guys. Did I already do all this with you guys? When I talked about walking proteins, I've gone through all this.
No, I mean, you haven't went over, you just like briefly.
So I briefly hit it. Okay.
Yeah, like how it moves.
Okay, so we're right where we are. Okay, so we're in the right spot. Just making sure. You have me confused for a second. So cytoskeleton. It's made of three main components. Our microtubules, our microfilaments, and our intermediate filaments. The microtubules, microfilaments are very active in movement. They're not these static structures. There's these dynamic structures that help to control the shape of our cell membrane. The intermediate filaments are really anchorage points. They're more rigid structures. They're there for anchorage. So when we look at cytoskeleton, we look at these three main components of it. You don't need to memorize any of this stuff. This is just kind of FYI. What would I know about microtubules? I know that they're kind of the largest of the three in terms of diameter. And they really help with cell motility, cell movements, organelle movements, cell shape, right, all that kind of stuff. So they help the cell move. They can make up cilia, flagella. They can help maintain the cell membrane shape. When we think of mitosis, right, at the end of telophase, this other process begins. Starts with a C. Nice and loud so we can hear you on the video. Cytokinesis, right, where the cell membrane starts to pinch off, right? Well, what controls the pulling in of that cell membrane? How is the cell pinching itself, forming these cleavage furrows? Well, it's the microtubules and microfilaments of the cytoskeleton that are pulling on the cell membrane, causing its shape to change. So the microtubules, not only with cell motility in terms of cilia and flagella, but they also maintain the cell shape, structure, and these are also the monorail system, right, where we're moving vesicles within the cell. Everybody okay? Microfilaments, right, with microfilaments, we're looking at the thinner fibers, but these are also part of maintaining cell shape. When we talk later on about our muscles, right, if everybody makes a muscle, right, what happens, you have a whole bunch of filaments contract on one another. Those filaments are actually these little microfilaments, these actin filaments. So this is also involved with your muscles as well, kind of cool. But just how you have kind of that sliding action in your muscles, you get the same kind of sliding action in your cell, to pull the cell membrane in, to push the cell membrane away, and so forth. So this is involved with maintaining cell shape. With that, like an amoeba, everyone knows what amoeba looks like, kind of that odd-shaped cell. Can really change its shape to many different forms. Well, it's the microfilaments and microtubules that allow that amoeba to kind of morph its shape. So we have two of the real active forms, right? And then we have our intermediate filaments. This is really kind of the inactive form. This is really the structural component. When I want to anchor something to a cell membrane, or I want to make sure I'm just being rigid with my cell shape, that's where these intermediate filaments come in. They're not really active like the other ones. They're not dynamic. They're more static. Okay, so those are the three main components of our cytoskeleton that controls our cell shape. Those microtubules, though, also play a role with a few other things. So this is an amazing picture, right? A tunneling electron microscope transmission, electron microscope. So we actually see the cytoskeleton of a cell. I know, it blows your mind, right? It's amazing. Cytoskeleton. You woke up today going, I wanted to look at the cytoskeleton of the cell. So we have our microtubers right here. We have our microfilaments, all components of the cytoskeleton. And then this is what I was talking about the other day, is how that microtubule can act as the rail system for movement of vesicles. So this concept we spoke about yesterday, the other day, we're going to kind of relate it to how cilia and flagella operate as well. Okay? So just kind of remembering this, right? This protein kind of holds its shape. How do we get this motor protein to change its shape? What do we do to it? Al, do you remember? Trying to. Remember? What do we do? We definitely talked about this the other day. 95% of the time, the answer's on board. What's ATP? Energy, right? We energize it. ATP comes in. ATP energizes it. When I say ATP energizes something, what is it actually doing? It's changing its shape. How is it changing its shape? What is ATP actually doing to it? Giving it a what? It's giving it energy. It's giving it charge. Electrons are involved because we're breaking or forming bonds. I am out of markers. So when I say ATP, I'm saying adenosine
triphosphate.
and adenosine triphosphate, right? So here's my 5-carbon sugar. Here's my nitrogenous base. There's our ATP molecule, adenosine triphosphate. How does ATP energize something? How does it give energy to something? It gets its energy from sugar, right? Remember, sugar is like a $10,000 bill that we can't use directly, so we break it down into usable energy, which is like the 5s and the 10s with ATP. So what is ATP? This is like a really big picture thing. This is a good thing to understand. What does ATP actually do to energize another molecule? Energy is not this abstract thing. Energy is very concrete. What types of energies do we have out there?
Light energy.
Moving energy, what's that called? Kinetic energy, right? What about this? What type of energy do I have now? Potential energy, right? Gravitational potential energy out there. Another type of energy, what happened?
I thought you were going to fall. I got really scared.
What other type of energy? Heat energy, light energy, right? Solar energy. The one we're focused on, though, is inside. Like when you eat food, what type of energy are you getting? I'm not getting heat. What is chemical energy? Where do chemicals hold their energy? Nice and loud so they can hear you on the video. Bonds. In the bonds, in the chemical bonds. That's what chemical energy is. Glucose has all of this energy in all those chemical bonds. So much that your body can't use it directly. We need to break the energy out of those bonds to put it in a whole bunch of ATPs. So, where does ATP hold the energy? In the bonds, but specifically what bonds? ATP gets broken down into ADP. So therefore, where's the energy? In that bond right there. That's the energy. So if ATP were to transfer that phosphate, it would be giving whatever picks up that phosphate energy. So with our walking protein, ATP energizes it. It's basically saying that ATP is doing what? It's giving that protein energy. It's giving that protein a blank group, a phosphate group, right? And when I add a new chemical bond here, right? When you have a chemical and you put a new bond on it or you break a bond, what does it do to the shape of that chemical? Changes the shape. Protein is just a chemical. It's just a macromolecule, so it changes the shape. So when I add the ATP, the shape changes in one direction, takes a step forward. When I dephosphorylate, when I take the ATP off, it goes back to its original shape, balances out. So ATP is involved here, kind of powers this process. Okay. So we kind of, the basic understanding of this right here. Now it's going to be useful as we move forward. Let's get this out of the way before we really get into the actual movement again. But just a couple key structures that we should be familiar with here are centrosomes and centrioles. What process are centrosomes and centrioles involved with? Nice and loud. Mitosis. They're both involved with mitosis, right? Everyone remembers mitosis, right? PMAT, prophase, metaphase, anaphase, telophase. What structure or what structures originate from the centrosomes and centrioles? Microtubules, and another word for it. When we talk about mitosis, we didn't really use the word, or you most likely didn't use the word microtubules. What word did you use? Next to the centromeres, the chromosomes, those little things. Spindle what?
Fibers.
Spindle fibers. And remember spindle fibers? Right, from mitosis? Guess what spindle fibers are made of? Microtubules, and they originate from this region. See how good you guys are remembering these from way back when. Centrioles. Do you find centrioles in animal cells or plant cells? Animal cells. Plant cells do not have centrioles. The centrioles are just kind of like an anchorage position. The real working is the centrosome, which is the region. So you have this to kind of take a look at on your own. So here are my centrioles located in the middle of the centrosome. So where your spindle fibers are put together, where your microtubules are arranged into your spindle fibers, that area of the cell are known as centrosomes. That's it. It's really where the work's being done. In animal cells, we have these two kind of little anchorage points known as microtubules. In plant cells, you just don't have them. They're just kind of originating there in space in that centrosome region. Kind of a side note, but it's here. They're made of the same structures. Everyone okay with centrosomes, centrioles? Just basic stuff? Okay. All right, so now let's get into what we really want to talk about today, which are cilia and flagella. Cilia and flagella are simply microtubules control the beating of cilia and flagella, locomotor appendages of some cells. Which one's larger? Flagella, cilia are typically smaller. When I have a cell with flagella, is it a numerous number or a small number? A larger number of flagella or a smaller number of flagella? Smaller flagella, more cilia. As you'll see kind of, I think, in the next picture right here. So here is an example of a cell with a flagella. That is a sperm cell. And then down the bottom here is an example of a cell with cilia. This could be a bacterial cell. Could be an example. You can see how many cilia are there. They both operate in a very similar way, which we'll talk about in a second. But there's a little bit more complexity with the movement of the flagella. It works more in a rotational movement instead of just a straight back and forth. Okay. Now, for people with SAT2s, this is kind of a big SAT2 question. Don't worry so much about the number, but in SAT2, they like the 9+2 pattern. It relates to the pattern of microtubules within a cilia or flagella. So let's just kind of understand what we're looking at here. Here's a plasma membrane. Here's a cilia or a flagella coming out of the plasma membrane. So where they stay, it's calling it a cilium. So this is a cilia right here. Their structures are the same. It's how they move that's a little bit different, the coordination. So in the base of your cilia flagella, you kind of have your anchorage point, and that anchorage point is known as a basal body. It's really just kind of an arrangement of microtubules that help to anchor it into the plasma membrane. Coming out of it, you have your cilia or your flagella, and the microtubules are in a 9+2 pattern. What it's saying is there are 9 pairs of microtubules around a central pair. That's the 9+2. That's what it's referring to. Okay, and this is basically what the structure looks like. And the other thing that we have to kind of notice here are these little red dashes. These are the actual proteins. So like we had our walking protein, our motor protein. These are similar to that. They're called dynein proteins. And I'll give you that word. I think it's on the next slide, but just in case. Dynein proteins that aid with movement. So relating how ATP actually causes movement. This is good for you to read on your own. So this is basically what I'm about to say, a brief synopsis of it. So now we've broken it down. Instead of looking at the entire ciliar flagella, we're just looking at 2 pairs of microtubules. So instead of looking at this whole thing, we're just looking at these 2 right here. We just simplified it a little bit, but you can picture it happening with all of them. These are proteins. What happens to proteins when they're phosphorylated, when they're energized? They simply do what? They change their shape. That's it. So when I energize these proteins, they change their shape. Now these proteins are anchored from one microtubule pair to the next. So when they change their shape, what do they cause the microtubules to do? Bend. That's it. Just like the walking protein, it's that simple. That's what we're talking about with energy and ATP, right? Conceptually, this is how our body works, right? So these are 2 great examples to talk about what ATP does for us. Instead of just being this vague, it's energy in our body. This is how it works. So ATP comes in. ATP adds a phosphate group to all of these dynein proteins, causing them to change their shape slightly, causing the cilia or flagella to bend. Then the phosphate group is released, and the proteins go back to their original shape. That's all it is. That's how this process works. One last thing with this is just getting back here. So the only difference in the process between the flagella and the cilia is the flagella movement is just a little, the cilia movement's a little less coordinated. It's kind of like all or none, just back and forth. Where with the flagella, you kind of get this progression of phosphorylation. So they're not all phosphorylated at the same time. You get them phosphorylated in different regions, causing it kind of to get this spinning type motion. Not that you need to really differentiate the 2, but that's kind of the main difference between them. Great questions? Pretty straightforward? Not too bad? Yes?
Does it always bend in the same direction?
Does it always bend in the same direction?
Like if it's phosphorylated, then is it always, like with that picture, it's always going to the right? Or does it?
That's a great question. I don't know the answer to that. Another question, instead of bending in the same direction, does the flagella tail always spin in the same direction?
Yeah.
Does it always spin clockwise or counterclockwise? I don't know. It's a good question. It's something to look up. My guess is that there is a set pattern. So if it spins counterclockwise, my guess is it's always spinning counterclockwise. Would be my guess, but I'm not sure. Okay. So just kind of looking at other movements within the cell dealing with these microtubules and microfilaments. One is, I kind of mentioned it before, the amoeboid movement, or the amoeba. Right, so here's kind of our amoeba cell. There's like that shape-shifting cell, kind of forms all these weird kind of blob shapes. Right, so all these microfilaments and microtubules can help kind of extend the cell membrane, extend the cytoplasm in one direction. So we can kind of grab on here and then slowly slide its body over. Just controlled by the microtubules and microfilaments. The one last thing that really kind of plays a role with our cytoskeleton and movement is cytoplasmic streaming. Not everything in your cells get moved from one area to the next via some type of microtubule and vesicle and motor protein. Some materials are just dissolved in your cell, in your cytoplasm. So how do we move dissolved oxygen, carbon dioxide, dissolved nutrients? How do we move it around the cell so it doesn't stay concentrated near the cell membrane? We do it with cytoplasmic streaming. How many people have ever been in a round swimming pool, bunch of friends, start going around the outside, create like a whirlpool? Who's done that before? 1, 2, 3. Oh, people haven't lived. It's so fun. We used to do it growing up in a bunch of friends at above ground pools. It's always better in the round above ground pools. But in any case, you start moving around the outside, you create this current. Cells do the same thing. Slight vibrations or fluctuations in the cell membrane. This is a plant cell, so there is a cell wall, so it's only so much flexing. But by pulling on the cell membrane, you start moving fluids, and doing it in a rhythmic type fashion, it can kind of create these currents inside the cell, just known as cytoplasmic streaming. So how can cells move materials that are not in vesicles around the cell? Well, they do it via cytoplasmic streaming. And this isn't anything big right now, but just kind of mentioned it. I did talk about before with the muscle. The actin and myosin filaments, the actin microfilaments, it's made of a thing called actin. They are involved with muscle contraction. So don't worry about this. This is just kind of mentioning it, relating that the same structures that cells move for motility, those same structures still work for organisms using it in terms of muscle contraction. So just kind of showing it, but we'll talk about that towards the end of the year. Okay, as if that wasn't exciting enough, right, we need to talk a little bit about the ECM, the extracellular matrix. The ECM is really kind of this connective material outside of cells, and it involves a number of different structures. It involves a bunch of carbohydrates, a bunch of proteins, and all these different connections. So we're going to get to the ECM, but before we talk about ECMs in animal cells, we're just going to mention just a little bit briefly about the cell wall in plants. There's a little bit more to a cell wall than just straight, it's a cellulose wall. There's a little bit more complexity there. Nothing too big, so we're just going to talk briefly about the cell wall. There are 3 main layers. You can ignore the 3 names. You do not need to memorize these 3 names. But what we want to do is understand that not all cell walls in plants are built the same. So here's kind of looking at our cell wall. So this is kind of the lamina, the area in between 2 cell walls. Here's what's considered the primary cell wall, which is the outer part, and here's considered the secondary cell wall. Now, the only thing I want to add to a cell wall, we know it's made of cellulose, we know it's for structural purposes. So really 2 key things I just want to kind of put out there right now. One, do with the question. Not all cell walls are created equal. And if we were to talk about the thickness of a cell wall, how would the thickness of a cell wall in a plant cell in a leaf compare to the thickness of a cell wall of a plant cell in the trunk of a redwood tree? Do you think those cell walls are going to be equal? Obviously not, right? Which one do you expect to have a thicker cell wall? The tree trunk. Obviously going to have a thicker cell wall. So by looking at thickness of cell wall, you can kind of see how plant cells differentiate their cells. When plant cells have more of a thinner cell wall, when that kind of secondary layer is very thin, most likely those are going to be the metabolically active cells. Those are going to be the cells in the leaves, in the green part of plants, undergoing photosynthesis. The thinner cell wall is going to relate to an easier exchange of materials, such as carbon dioxide, oxygen. A thinner cell wall is going to allow for the passing of light, the passing of red wavelengths, blue wavelengths, different wavelengths of light, and so forth. As you thicken that cell wall, it definitely becomes more structured, right? You don't really have photosynthesis going on in the trunk of the tree. That's why it's not green. There's no chlorophyll there. So that's just one kind of concept out there, is just kind of relating, not all cell walls are made equal. That's really it for now. But the other one is looking at these little connection tubes. One of my favorite biology terms is this thing known as plasmodesmata. Plasmodesmata are simply just connections between cell walls. If we had a completely rigid cell wall, the plant wouldn't function that well. Plant cells need to communicate with one another. They need to be an exchange of materials, exchange of nutrients, exchange of hormones, some type of communication. So how do plants allow for communication between cells? Well, they create these bridges or these tunnels that go between the cell wall to cell wall, and those tunnels are just known as plasmodesmata. Easy enough? So not too much in cell wall, just a little bit there. And this is a great picture. Sally, I knew you woke up this morning and wanted to look at the electron microscope image of a plasmodesmata. So I threw this one in here for you, just to kind of let you kind of so it's actually kind of a cool image, seeing the two cell walls and then seeing the actual chambers between them, allowing for the passing of nutrients, the passing of hormones, and so forth. All right. The extracellular matrix, right? Our cells, they do not operate. Multicellular organisms, their cells do not like to be on their own. Those cells like to be connected to other cells, hence they're multicellular. So the mechanism to connect them is known as the extracellular matrix. So with the extracellular matrix, here's really what you need to know about it, is right here. It supplies support, adhesion, movement, and regulation. So when we talk about the extracellular matrix, we're going to look at some of the components of it, but really we just need to kind of know the function. The extracellular matrix, it acts for support, supporting one cell another, adhesion, holding one cell close to another cell, movement, not just movement of one cell moving along another cell, but also movement of materials, right? So the ECM can help guide hormones or food or nutrients from one cell to the next. And that guidance and that movement, it really is kind of a type of regulation, So support, adhesion, movement, and regulation. What is the ECM made of? Collagen, proteoglycans, fibronectin. You don't need those terms. You don't need to memorize those terms. But let's break down the one in the middle, proteoglycans. You should be able to tell me what proteoglycans are made of. Proteins and? What are glycans? Glycogen. Which is a? Sugar, carbohydrates, right? Carbohydrates. So proteoglycans, glycogen is a carbohydrate. Proteoglycans are materials, components made of proteins and carbohydrates. Just a good word to be able to break down, proteoglycans. Not that you need to memorize that they're all made of collagen and fibronectin, all this stuff, but understanding what that word means is a good thing. So here we're looking at our ECM, our extracellular matrix, right? And we just have a lot of different components, right? We have proteins, we have carbohydrates, again, connecting one cell to the next, allowing for the movement of materials, and so forth. Here's just kind of a close-up of a proteoglycan. Again, it's just a simple image, but just showing that you have the protein core with the glycogen, the carbohydrates coming off, hence the word proteoglycans. So we will finish right there. This class is over.